Module 6, Chromosome, Macromolecular Organization, Primary and Secondary Construction, SAT bodies and telomeres. The main objective of this episode are to know the history about chromosomes, to analyze the construction in chromosomes, to understand about the telomeres, to know about the macromolecular organization. In 1888, Waldeyer introduced the term chromosome for the presence of the color body in the eukaryotic cell. In 1916, Calvin Bridges proved that chromosomes are the physical carriers of genes. It is made up of DNA and associated proteins. Each DNA molecule that constructs a linear chromosome must have a centromere, 2 telomeres and the replication origin. This module can be discussed under the following 4 subheadings. They are A. History, B. Macromolecular organization of chromosomes, C. Construction in chromosomes, D. Telomeres. The chromosomes in initial meiotic prophase shown morphologically distinct regions which were consistent in size and position. These regions are called as chromomeres. Chromomeres are bead-like bodies over the chromonemata. Chromomere takes up more significant amount of stainable nucleoproteins. Chromomeres are regions of the superimposed coils accepted by the electron microscopic observation. In 1928, Belling observed 1500 to 2500 chromomeres in a haploid set of chromosomes of ileum and alloy. Each chromosome of metaphase consists of 2 symmetrical structures, the chromatids, each of which contains a single DNA molecule. The chromatids are attached only by the centromere and become separated at the start of anaphase when sister chromatid migrate to opposite poles. Metaphase chromosomes have 2 chromatids, whereas anaphase chromosomes have only 1 chromatid. The functional unit of chromosomes is chromatid in crossing over cell division and in cell segregation. Chromosomes are formulated into large domains called chromosome territories. Chromosomes exhibit distinct levels of compaction, with the most condensed region indicated as heterochromatin, which is lacking in genes or consists of genes that are transcriptionally suppressed. Less condensed region of chromosomes are called euchromatin, which is rich in gene concentration and is usually, but not always, actively transcribed. The network of fibers that take up the stain in the presence of aceto or syn, basic fuchsin, is called chromatin network. At the interphase stage, chromosomes are relaxed and diffused. Such chromosomes emerge as a network of fine chromosomal fibers. However, during the cell division, such diffused chromosomes undergo spiral coiling and condense into cytologically visible compact threads. At the metaphase stage, the number of chromosomes can be counted and its morphological features can be made out. Metaphase chromosomes are made up of two sister chromatids which are highly coiled. A chromosome is a hereditary material, a copy of it passed on to each daughter cells at the division. Each chromosome can replicate and newly replicated copies are subsequently be separated and partitioned correctly into the two daughter cells. In the nucleus of an individual cell, the DNA molecule is bundled into thread-like structure known as chromosomes. Each chromosome is made up of DNA firmly coiled many times over proteins called histones that supports its structure. The DNA that makes up chromosomes grow into more tightly packed during divisions and is then visible under a microscope. Chromosomes, when viewed through an optical microscope, appear as highly coiled double-stranded threads. In the haploid sets of chromosome, each of the chromosome is genetically and morphologically distinct and characteristics, while the number of chromosomes that constitute a haploid is constant in species, however, varies from species to species. Two sets of haploid chromosomes are formed, a diploid set. Thus, chromosomes are considered as vehicles of heredity and act as the repository for genetic information required for an organism. Suspended within the nuclear sap is a network of threads. When stained, they take color. Hence, they are called chromatin threads. Some of these threads are associated with either pore complexes or inner nuclear membrane. The interphase chromatin network is not a constant feature, but changes as and when the cell possesses through various stages of cell division. When the chromatin at the G1 stage appears to be diffused, thin, single stranded and coiled, however, as the cell division proceeds, the chromatid undergo duplication to form double stranded chromatin threads. During this process, DNA replicates and the histones and the non-histones are drawn from the nuclear sap to form sister chromatin threads. From interphase to prophase, a long DNA threads undergo continuous coiling and condensed into thicker structure. At the same time, the chromatin disentangles from the network and the chromosomes slowly get resolved into individual threads. These threads are referred to as chromatids. If such chromatids are carefully observed under 2000 times enlarged high resolution light microscope, each of them appears to be spirally coiled. The chromosomes show differential condensation and some regions take more stain and other regions take less stain. The darkly stained region is called heterochromatic segments, While the lightly stained regions are called euchromatic, this differentially staining behavior is called heteropyknosis. The tightly coiled heterochromatic segments are named as positive heteropyknosis, whereas the less coiled is called as negative heteropyknosis. The detailed aspects of heterochromatin can be studied in the next topic. The inheritance of Mendelian factors through chromosomes was not substantiated until the unit character, that is gene inheritance, primarily located within chromosomes. However, the problems of genes which are arranged in chromosomes was solved by the discovery of bead-like structure in meiotic chromosomes called chromomeres. At leptotin stage, the chromosome appears as subtle strings of beads. Further, chromomeres were named as the coiled expression of chromonemata, which may disappear when two ends of such chromatids are stretched apart. Later, these experiments helped to build the concept as the gene is a unit of heredity, a unit of mutation, a unit of recombination, and a unit of function. Particular types of chromosomes are found only in specific individual tissues. These chromosomes are more extensive and are called giant chromosomes in individual plants, and they are found in the suspensor of the embryo. The Paulatine chromosomes and Lambras chromosomes occur in animals and are also called as giant chromosomes. Chromonema fiber is a chromatid fiber 100 to 130 nanometer in diameter, thought to be an element of higher order packing of chromatin within the chromosome. During prophase, the chromosomal material becomes visible as fragile elements called chromonemata, which is called as chromatids in early stages of condensation. Chromonema and chromatid are the same structure where linear DNA molecule associated with proteins. Chromomeres are bead-like structure visible in interphase meiotic chromosomes. They can be seen in Paulatine chromosomes. At metaphase, they are not visible. At the metaphase stage, the number and the detailed structure of them can be studied with a light microscope or electron microscope. Many chromosomes vary from organism to organism, minimum 2 chromosomes to 1600 chromosomes, and this number is constant and characteristic for a given species. For example, Drosophila melanogaster has 2n equals 8, human has 2n equals 46. The haploid set consists of one set of chromosomes, and each chromosome is different from other structurally, genomically with unique characteristics. The terms triploid, tetraploid and polyploid indicates the number of sets present in the nucleus. Generally, organisms have a constant chromosomal number. However, sometimes vary in chromosome numbers due to gain or loss of chromosomes. In sometimes the entire set of chromosomes is involved. These variations ultimately lead to the origin of species, which is one of the fundamental steps in organic evolution. Chromosome size and shape. Although chromosomal size varies from organism to organism, a particular size of chromosomes is constant for a given species. In an organism, all the chromosomes are not of the same size. Some chromosomes are called giant chromosomes, for example, Lambrecht chromosomes in Genus Levis, salivary gland chromosomes in Drosophila melanogaster, and chromosomes in the endosperm, Haustoria of Pachyolus are 100 to 1000 times larger than their somatic chromosomes. These are called a particular type of giant chromosomes. Almost all chromosomes are spirally coiled, thread-like structure, but at the anaphase stage, chromosomes show a specific bent shape. This is because of the position of primary constriction or centromere. The V-shaped chromosomes are called metacentric, J-shaped chromosomes are called submetacentric, and rod-shaped chromosomes are called acrocentric. Higher organisms like man and monkeys, where male and female sexes are morphologically distinguishable, have two types of chromosome. They are named autosomes and sex chromosome. The sex chromosomes are X and Y chromosome. For example, humans are with 46 chromosome with 44 autosomes and 2 sex chromosome. The autosomes are for the development of physical body, and the sex chromosomes are generally responsible for the expression of their characters. If 2 XX chromosomes are present, the female character is expressed. If 1 X and 1 Y chromosomes are present, the male character is expressed. The X chromosome is considered to show the female characters and the Y chromosome for the male character. Y chromosomes are heterochromatic and X chromosomes are euchromatic. Further, sex expression varies in different organisms. In total, the interaction between autosomes and sex chromosomes ultimately determines the appearance of sexes through the mediation of specific female hormones estrogen and androgen, the male hormones. An individual chromosome general morphology depends on the position of this centromere, the dimensions of the chromosomes, and the presence of secondary constrictions and satellite bodies. 1. Primary constriction. A lightly stained region appears as a constriction of the chromosome and is usually called centromere or primary constriction. The centromere is the region where chromosomes get attached to spindle fibers. These centromeres lead the anaphase chromosomal movements. If direct X radiation hits destroy the centromere, the chromosomes behave abnormally and lose their directional movements. Electron microscopic studies clearly show 2 kinetochore structure in each centromere. The outermost region of the kinetochore are a cup-shaped structure called the commissural cup, and it is relatively thick. On its outer surface, a large number of processes called corona is present. The 3 to 10 microtubules are found to be penetrated to each chromonema. Based on the centromeric position, chromosomes are classified as follows. 1. Metacentric, where the centromere is present in the middle. 2. Submetacentric, where the centromere is present little away from the middle. 3. Acrocentric, where the centromere is present at the extreme tip, but with a small segment at the end. 4. Telocentric, where the centromere is present at the extreme tip of the chromosome. If 2 or more centromeres are found in the same chromosome, they are called dicentric or polycentric. In holocentric chromosome, the centromeric activity is distributed throughout the length of chromosomes. This kind of chromosomes are seen in C. elegans and many insects. Another exciting feature of the centromere is that on either side of the centromere, a chromatin material is heterochromatic. Microtubules are attached with the centromere. It helps in chromosomal movement during cell division and the formation of spindle fibers during metaphase division. At the same time, acentric chromosomes result due to abnormalities which are induced, for example, by X-ray in chromosomes and are unstable and cannot attach to the mitotic spindle and remain in the cytoplasm. 2. Secondary construction. Along with the primary construction, another construction is present on only 1 or 2 pairs of specific chromosomes. Such construction is called secondary construction, and hence it is characterized by its nucleolar formation. It is also called a nucleolar organizer. The secondary construction exhibit a negative heteropyknosis which contains DNA segments for cytoplasmic ribosomal RNA. The genes present here are abundant or tandem repeats. In some cases like frog oocyte, these genes get amplified at the end of the telophase. The DNA in this region opens out and start transcribing ribosomal RNA as abundant precursors. This RNA is sliced and processed into 28S and 5.8S RNAs. These RNA in turn get associated with different ribosomal proteins subsequently, and functional ribosomes are produced. If the secondary construction segment of the chromosome is knocked off, the nucleolus fails to appear and the cells die prematurely. 3. Satellite bodies. The region of chromosome which is present beyond the secondary construction is called the satellite body. If the secondary construction is very close to an end of the chromosome, this satellite may be seen as perceptible dot. Only in human 2 chromosomes possesses such a zone, and hence they are called as the nucleolar chromosomes. Chromosomes bearing satellites are called SAT chromosomes. SAT means acido-thyminonucleolico. Generally, one chromosome in each genome possesses this satellite body. There could be 2 or more satellite bodies in a single chromosome. The extremity of chromosomes are usually called telomeres. The term telomere is coined by Muller in 1938 to indicate its uniqueness. The intact end of a chromosome generally does not enter into permanent association with any one chromosome. The loss of telomere loses its stability to the chromosome to which it was once attached. The telomeres is the terminal part of the chromosome. It offers stability to the chromosome. DNA of the telomere has a specific sequence of nucleotides. Maintenance of telomere appears to be an essential factor in determining the lifespan and the reproductive capacity of cells. Therefore, studies on telomeres and telomerase have the promise of providing new insight into conditions such as aging and cancer. Telomeres prevent the fusion of chromosomal ends with one another. When X-rays break chromosome, the free ends without telomeres become sticky and fuse with other broken chromosomes. However, they will not fuse with the normal telomeres. Telomeres at the end have a long linear DNA contained in each chromatid. The sequence of nucleotides in telomeres is AGGGTT, with the complementary DNA strands being TCC-CAA, with a single stranded TTA-GGG overhang. This sequence of TTA-GGG is repeated about 2500 times in humans. In humans, the average telomere length is about 12 kilobases at birth. In the old age, it reduces to 4 kilobases. The reduction of telomere is more significant in males than females. The eukaryotic DNA replication enzymes cannot replicate the sequences present at the ends of the chromosome. This is called as end replication problem. Therefore, telomeres are successful in cell division. They cap the end sequences and they got lost in the process of DNA replication. Interestingly, the cell has an enzyme called telomerase, which adds the repetitive nucleotide sequences to the ends of the DNA. Telomerase thus replenishes the telomere cap of the DNA. Generally, in higher organism, telomerase is active in specific white blood cells, germ cells and embryonic stem cells. Telomerase can be reactivated and reset back to an embryonic state by somatic cell nuclear transfer. The shortening of telomeres with each replication in somatic cells may have a vital role in senescence and the prevention of cancer. This is due to the loss of telomeres after a certain number of cell divisions and loses the essential genetic information from the cell's chromosome with future divisions. To conclude, chromosomes are hereditary materials. They play a dynamic role in cell division. Thank you.